So now we're going to talk about the environment and microbial growth. This is split into two parts, Part D and Part E. So the environment significantly impacts microbial growth. We're going to consider temperature, pH, osmolarity, and we're not really going to talk about pressure, but we are going to talk about, I missed a few things on this slide here. We are going to talk about oxygen status. So we're going to talk about this, this, and this. We're not really going to talk about pressure, but I do want you to be aware that it matters. And we're not going to talk about neighbors, but I want you to be aware that that matters too. What other microbes are living in the environment will often dictate how many resources and whether there are toxins present or not. So they're signaling one another and they're communicating and deciding whether to grow rapidly or not to grow rapidly. So I'm not going to talk about those factors, but they are important. Let's think first about temperature. So temperature has profound impacts on organisms, including microorganisms. As temperature rises, chemical reactions, including those catalyzed by enzymes, happen at more rapid rates. This makes growth rates increase as well. Once temperatures reach a maximum temperature tolerated, and then those temperatures go beyond that point, however, you see a rapid change in the situation. Proteins will denature and cellular macromolecules can degrade, causing cell death. And that's why we see this sharp plummet. So you see this smooth increase as reaction rates are increasing, and that causes growth to increase proportionally, but then you hit a particular temperature and then bam, it falls off completely. It's not a bell-shaped curve. So once temperatures reach a maximum temperature, then they will, as I said, proteins will denature and you'll see cell death. That's what's happening in here, is death. Okay? So therefore, for each organism, there are a set of what are known as cardinal temperatures. That means a minimum temperature at which the cell can grow at all, an optimum temperature where growth rate is the highest possible for that organism, okay, and then a maximum temperature beyond which you will see protein denaturation, collapse of the cytoplasmic membrane, thermolysis, and death. So those are the cardinal temperatures, and they will be unique for each particular species or even subtype of species found in different environments. Let's think a little bit about the adaptations that are involved in helping organisms deal with temperature extremes. But first, actually, let's talk about how microbes are classified based on temperature and their growth response. So a psychrophile, actually let me back up. The term phile approximately means to love. Okay? So a psychrophile is something that loves cold temperatures. And here's an organism, and here are its cardinal temperatures. There's its minimum, its optimum, and its maximum. And look how cold it is, right? Remember that 0 is freezing. All right? So this is something that can actually grow pretty well at temperatures below the temperature that freezes water, and it grows optimally at 4 degrees C, which is the temperature of your refrigerator. And in fact, as soon as it gets warmer than a typical wintry day in New Hampshire, it won't grow at all. So this particular organism, Polaramonas vacuolata, is from Antarctica, and it much prefers cold temperatures, and in fact it cannot tolerate even moderate temperatures. Room temperature would be way too hot for that organism. A psychotroph, in contrast, it will tolerate cold temperatures, but it grows best at moderate temperatures. Okay. Mesophiles are organisms, these would be the organisms associated with the human body and also many other environments. So for example, E. coli. It can grow at colder temperatures down here around 10 degrees C, but its optimum is close to 37 degrees C, which is the temperature of most warm-blooded mammals. The temperature extreme falls off somewhere at, around 48 degrees Celsius. That's the maximum. Okay. Then we're up into the thermophile world. So thermophiles like it hot, hyperthermophiles like it really hot. So we've got thermophiles that have temperature optimums around 60 degrees Celsius. That's pretty warm. And then hyperthermophiles, and we see two profiles here. One for a bacterium, Thermococcus celer, and then another for an archaeon, Pyrolobus fumari. The extreme hypothermophiles, those that can tolerate temperatures above the boiling point of water, which is here, right? That's where water boils. So only in environments where you have high pressure do you see these circumstances. High pressure allows for temperatures that exceed the boiling point of water, and if we look at some of those environments, we see specialized, highly adapted archaea that are able to actually not just tolerate, but survive and thrive. So it's important to note that these are growth profiles shown here. So these organisms are not just tolerating these conditions, they're actually growing well under these conditions. So many of the psychrophiles that have been isolated are from the Arctic and the Antarctic, places that are constantly cold. For example, there are lakes in Antarctica, the McMurdo Dry Lakes, and McMurdo Sound is an area from there. McMurdo Sound is on the coast, but there are lakes called in the McMurdo Dry Valley. And if you look at these lakes, and one is shown here, there's a constant ice that is meters thick. So that's a fabulous place to look for psychrophiles, and in fact many have been isolated from there. Many polar ammonis species of bacteria and a variety of algae and diatoms have been pulled up from Antarctica and also from the Arctic, and these are true psychrophiles. They grow optimally at very cold temperatures. So what allows them to do this? If you look at the molecular adaptations that psychrophiles have, they can be described as three major adaptations. They have different phospholipids, they have cold adapted enzymes, and they have cryoprotectants and cold shock proteins. So their phospholipids tend to have relatively short chain fatty acids and a much greater degree of unsaturation. In fact, there have actually been microbes that have been isolated from Antarctica that make what are called polyunsaturated lipids, the only ones found on Earth that can do that. So if you have a higher degree of unsaturation and relatively short chain fatty acids, you look like this. Okay, you have very sort of kinky fatty acid tails, and they don't pack together well, and if you look at them in the membrane as shown here, the membrane will be not as well packed and it will remain fluid at very low temperatures. Okay, if you look at the enzymes that these organisms have, they're cold adapted, so they have particular amino acids. They tend to be highly polar, use more of the polar amino acids in a lot of their enzymes, and they seem to function much better at cold temperatures. They also make molecules like glycerol and other molecules to keep ice from crystallizing within the cell. Once ice crystallizes in the cell is dead. So cryoprotectants function like antifreeze basically. So these are often molecules like glycerol, as I said, or often proteins that will help to serve as cryoprotectants. And cold shock proteins that work to coordinate gene expression under cold conditions, and that would be the expression of genes required to make the cryoprotectants. So those are the adaptations that contribute to what is called psychrophily, or the ability to grow at very cold temperatures. All right, let's think about the opposite end of the spectrum, those who like it hot. So let's think about thermophily and hyperthermophily. And let's first consider the optimal growth temperatures for various organisms that are found on Earth. So this table depicts the upper known temperature limits for the growth of many different types of organisms. You'll note that animals and plants are pretty wimpy in this regard, and they just do not tolerate particularly hot temperature, at least the ones that are shown in this table. In contrast, there are many microbes that can tolerate temperatures at which water boils or above in highly pressurized environments. These tend to be bacteria and archaea. The eukaryotic microbes are not as high temperature loving. So here we've got a variety of protozoa, algae, and fungi. These are eukaryotic microbes, and they're tolerating, warm temperatures but not truly hot temperatures. Where if we look down here, there are many known archaea that can tolerate temperatures that are higher than the boiling point of water, and some bacteria that can tolerate very high temperatures as well. An example of a place where you might pull out either bacteria or archaea might be Yellowstone Park, kind of a mecca for microbiologists. The hot springs there have been sources of numerous thermophilic and hyperthermophilic organisms. These might come from deep sea hydrothermal vents, places where it can get hotter than 100 degrees Celsius in the water because of the high pressure. Okay, so thermophiles have unique molecular adaptations too. They have very heat-stable membranes. For example, you've already looked at the monolayer membranes seen in thermophilic archaea. They also tend in the bacterial world to make highly saturated fatty acids in their phospholipids, the opposite of what the psychrophiles do. And they'll place a lot of what are called hopanoids and other stiffening agents in their membranes to help to keep them from dissolving apart under high temperatures. They make heat-stable enzymes, and interestingly, those enzymes aren't a lot different from what we see in mesophilic organisms. They'll just have a few very important amino acid substitutions that seem to confer the ability to function well under high temperature. And they'll make solutes that protect proteins at high temperature. So here's a question for you. What is an essential heat-stable enzyme that revolutionized science, and what organism did it derive from? It's something that you should remember from genetics or your other classes, so I'm going to leave that as a question for you. All right, let's think a little bit about acidity now, as measured by pH. So remember that pH is a logarithmic function of the hydrogen ion concentration. So going from pH 7 to pH 8 means a 10-fold increase in the amount of hydrogen ions, or the acidity. So when you go from pH 7 to pH 8, or similarly from pH 1 to pH 2, then that equals a 10-fold increase in hydrogen ion concentration. Okay, that's a lot. So 1 unit on the pH scale is significant. It's a significant change in concentration. So there are many environments in nature that have relatively extreme pH. If you think about your mouth over the course of a day and the fact that there are many acid-producing bacteria in it, while their overall mouth pH is fairly close to neutral, there are going to be pockets on your teeth and at the gum line where the pH is actually quite acidic, where it could get down to 4 or perhaps even 3, depending on how long it's been since you brushed your teeth. Okay? Similarly, there are environments in nature that are quite alkaline. Soda flats found in a variety of locations on Earth are highly alkaline. Also, humans have created environments that have extreme pH through the production of materials. For example, battery-making plants can have wastes that are down at this extremely alkaline end of the soil, or acid mine drainage, which comes out of a lot of open pit mines and other types of mines, can be pH around 2. And then also food has a wide range of pHs in it. So there are a lot of environments that have a wide variety of pHs, and microbes have evolved over time to tolerate those pHs and to in fact thrive at them. So we describe microbes according to their ability to thrive under various pH regimes. We say that neutrophiles are what like to grow in environments where the pH is greater than 5.5 and less than 8. So they ideally love 7 or neutral pH. E. coli is in that category. Acidophiles like it acidic, so they like pH less than 5.5. Here are examples of a few different organisms that fall into that group. This particular one is common in acid mine drainage. In fact, it's one of the only environments that it's been isolated from. Alkalophiles like pH greater than or equal to 8. So there are quite a few known bacteria that fall into that category. So one of the things that's really interesting is that neutrophiles, okay, this category of organism here, they will maintain an internal cellular pH, okay, in their cytoplasm that's about neutral. But studies of the acidophile and the alkalophiles, particularly the acidophiles, have shown that their internal pH is actually quite acidic. So it's something to think about. Another thing another thing to think about, and in fact there are a lot of riddles associated with some of the extremophiles, such as acidophiles and alkalophiles, is to question how they're running their proton motive force. Remember what a proton motive force is? It's an accumulation of protons, or H+, outside the cytoplasmic membrane in bacteria and archaea. And if you think about particularly an alkalophile, it's living in an environment where there's already a really low concentration of H+ on the outside. So given where it's living, how the heck does it manage to maintain a proton motive force? And that's really an interesting question, and I don't have the answer for you. Scientists have not yet figured that out.